Hello, good afternoon or good morning, depending on where you are, everybody. I'm very happy to give this presentation and I hope you'll find it interesting. I'm going to turn off the video just so we can get to the presentation and share my screen. So today the topic we're going to talk about is really practical approaches to studying energy balance and obesity. And with that, I'm going to tell you about this tool that we created and the problem that it solves, and we'll actually do an experimental run through. So for the talk, let's see, we have two parts. First, we'll talk about energy balance and CalR and how it can help. And then for the last part of the talk, I'll actually go live and we'll do a live analysis of some data using CalR with the internet from my home and with my kids in the background. So let's hope it all goes well and isn't too disruptive. Okay, so the very basics. Most of the people who would probably tune into this video are going to be familiar with energy balance. Energy in, under neutral energy balance, energy in is going to equal energy out. So that's very straightforward. And the energy in is going to be in the form of foods with different caloric values, different energy contents, and that is going to roughly equal the energy expenditure made up of resting energy expenditure, sleeping metabolic rate, arousal, the thermic effect of food, and activity-mediated energy expenditure. And these should sum up to the food intake plus whatever energy is not consumed and comes out in the feces. So we can use our modern indirect calorimeters, which will measure food intake, but they can also calculate indirectly the energy expenditure. And we can use a direct calorimeter, which will combust fecal matter to determine how much energy was not absorbed. So theoretically, this could not be any more simple. Practically, however, it does get a little bit thorny. So indirect calorimetry has been used in humans as well as in animals for quite a while. So people might be familiar with cycle respirometry, where you can try to get a maximal metabolic rate of an individual on a bicycle. There are chambers and rooms that humans can sleep in and live in to get their resting or activity metabolic rate. There was a pretty good story on that a few years back. And what we largely do is we analyze the metabolic rate of mice using these indirect calorimeters. Here's a 12-cage system that we have, part of our larger system. And you put the animal in the cage, measure its food intake. You have a pump which will measure the carbon dioxide that's produced and the oxygen that's consumed, goes into a computer, and you are indirectly assessing the energy expenditure by using some formula which have been empirically derived. So these are not derived from any first principles. These are formula that have been found to work over about 100 years now, and they've been tweaked several times, but none of the new variations are really dramatically different from the old variations. And the fundamental aspect is that you can calculate energy expenditure based on the rate of oxygen being consumed, the rate of carbon dioxide being produced, minus the amount of nitrogen which has been consumed in the diet, mainly in the form of protein. Since typically we'll be comparing animals on the same diet or with very similar amounts of protein, and measuring protein or nitrogen excreted is actually quite onerous, almost all the calculations that you see in the published literature, including the ones that we use for CalR, it ignores this one aspect, and we simply use an energy expenditure based on the oxygen consumption and carbon dioxide. We need to know this just in case we are, in fact, playing with animals with very different protein concentrations in their diets. You might see this in ketogenic diets or others. So it's a thing to take into consideration. But for most experiments, which is what we're talking about, we will use this very simple indirect calorimetry equivalent calculation. So when we're trying to do these calculations of get energy expenditure and compare energy expenditure in studies of obesity, there's a problem that we run into in the field again and again. And that comes from something people learn about in basic biology, in high school or college biology. And that is that bigger animals have greater metabolic requirements. And this was described in great detail in the 1930s, showing that you can compare a mouse and an elephant all by scaling the metabolic rate. And this is called a Kleber scaling factor. And back when he did it, which was quite a while ago, he had that the metabolic rate scales to body mass by a 3/4 exponent. And we now know on further studies for animals smaller than 10 kilograms, it's to a 2/3 exponent, and if you're greater than 10 kilograms, it's 3/4 exponents. So this is great if you have to compare an elephant and a man, or a dog and a goat. Those are great to know. But for the experiments that we are dealing with, we're talking about experiments of obesity, where you're comparing mice of different body compositions. And the Kleber scaling assumes similar body compositions among your guinea pig and your goose. So if you were to compare an obese goose to your guinea pig, then this would no longer be valid. So what we see is comparing a lean mouse to an obese mouse, while the total body mass does go up, the lean mass only goes up a little bit, and most of that increase in mass is due to fat mass. And fat mass is about 80% less metabolically active than the lean mass, which is why we can't do the scaling, and we can't just divide by total body weight, because none of these attempts to compensate for the larger mass are effective or are valid. So there was a great example of exactly this mistake published in Science in 1995, and this was sort of the first big impact example of how this could go wrong. The authors had beautiful data showing leptin had just been cloned, and this was a group at Amgen that had recombinant leptin, and they set out to show what the metabolic effect of treating leptin-deficient OB mice was with leptin. And you can see the body weight over just a few days decreases quite dramatically in this high dose of leptin, 10 mg per kilogram. You can see the body weight drops by about 20% in just a couple weeks. The food intake, as we now know very well with leptin, if you treat OB mice with leptin, their food intake drops about 50%, an extremely dramatic phenotype. So this data on food intake or body weight is not scaled in any way to body weight, it's just a percent. They also put these mice into a metabolic chamber, and they measured their carbon dioxide production and oxygen consumption. And if you see the effect of wild-type mice treated with leptin, there's no difference on their metabolic rate. But here, they're dividing by the weight of the animal. So they take the mils of oxygen produced, consumed, divided by the mass of the animals. And so they have a 20% decrease in body weight, and they divide the oxygen consumption by a 20% smaller number. So by dividing by a 20% smaller number, they've artificially increased their oxygen consumption by 20%. So this was, their data were fine, but their interpretation and calculations were erroneous. So they published their paper in 1995, and then 2 years later, Jean Hims-Hagen published a commentary saying that this analysis was likely incorrect. And the authors, to their credit, they responded and they said, you are correct, and if we analyze it that way, there's no difference of leptin on energy expenditure. And these studies have been done quite a while ago, and these have been replicated and even further confirmed in a more recent paper from Carl Kayala. You can read about it. But these, this was the first large-scale analysis where they did this wrong, and it was a high-profile paper. So if that had set the record straight, we probably wouldn't be having this conversation today. Instead, these two authors, Butler and Kosak, said there's a recurring problem where people keep doing what just happened in that Science paper. People had been inappropriately dividing by body weight when you have mice with more obesity or less obesity, and it was causing a series of artifacts. The way in which they did this was quite problematic. They made a table of everyone who they thought had done their studies improperly, called them out, and said that their data was not reliable. Their papers couldn't be trusted, and it was not particularly collegial. It was really rather a rough treatment of their colleagues. And a lot of people paid attention. I mean, the way that they got the attention wasn't great, but they were very effective in drawing attention to this problem in the field. In response, a bunch of authors put together a paper saying, a bunch of authors put together a response saying that an analysis should probably not be using dividing by body weight, but rather should be using analysis of covariance. And these are some of the leaders in the field, and this paper has been cited hundreds of times now and is widely being referred to. John Speakman put together a paper which was the 39 steps as an algorithm for performing statistical analysis on data and energy expenditure. So this was the computational solution to this recurring problem. People don't know how to do it. If you just run his simple program, you could find the answer very simply. And he provided data, scripts in Minitab, R, and SPSS. So this was just as I was starting my lab, and we had indirect calorimeter, and we looked to use this model and found it completely impenetrable. It was very difficult to follow this flow chart and to do all the appropriate statistical tests. And this is a perfect solution to a problem where if you are trying to examine any possible kind of experiment. So this is a really good idea, but practically not very useful. So that recurring problem that was mentioned by Butler and Kozak continued to recur. So you would have investigators like this one in JCI, where you would have mice with a 10% decrease in fat mass and body weight, and then divide once again by a 10% smaller number for the mass, and get this artificially increased oxygen consumption. You would also get statistics. So it's not clear what these stars mean. Is this for this day versus that day, or for this dark period and that dark period, not for this light period or that light period? Here's another example from Cell, where you once again have mice with lower fat mass of about the same amount as you get an increase in oxygen consumption rate. Here the statistics are even worse. They take any given point that is significantly different and they put a star over it. So they've done presumably a t-test at 24 different points, and they've not done multiple testing correction. Over here, more recently, I saw this paper just come out very recently. They have mice once again with differences in body weight, and here they're scaling to the 0.75 exponent, which would be appropriate if they were comparing two different species that were greater than 10 kilograms. So what this says is that when body composition is different, dividing by total mass is an invalid method of analysis. Complete time period should be compared, not individual time points. But what this says more than anything else is that investigators and students really need help with data analysis. This is a hard problem, and there really wasn't a tool out there to help people get through this. And this is complicated. So let's take an example right here. You've got two groups, a blue group and an orange group, and it looks like the orange group has a higher oxygen consumption during the dark, higher during the day. Looks pretty consistent, looks like really nice data. So you might be tempted to just take this plot, a bar graph of the means, and put a star over top and say, I have a significantly increased oxygen consumption rate, and move on with your data. But without knowing the masses, this plot could really mean three different things. If these energy expenditures, if this oxygen consumption was only because the animals were appropriately larger, 20% larger oxygen consumption and a 20% larger animal, then really there's no difference in the oxygen consumption when you take mass into consideration. It's also possible that, that these animals are the same weight and you do have an increased oxygen consumption, or that these animals are actually much, much greater in weight and have a relatively lower than expected oxygen consumption. So the solution proposed in that paper by Matthias Chopp and John Speakman and others, as well as analysis by Carl Kayala and others, suggested that using the ANCOVA, the analysis of covariance using mass as the covariate. So this one plot, depending on the mass, can give any of these three results, and the ANCOVA will help you differentiate. So in this one, you have two groups. This group does have a higher oxygen consumption, but it's appropriate for the larger mass. In this one, the two groups have similar mean masses and a greater oxygen consumption, which is significant. And in this one, this group does have a higher oxygen consumption, but given the much greater mass, is actually inappropriately lower. So there's a lot that goes into it, and you can see these very quickly and apparently when you use these regression-based plots and the analysis of covariance. So that's kind of the theme for the talk, that is a better way of analyzing and visualizing your data. To put it very simply, if there's one slide you take away from the whole talk, it would be this mean. Do not divide by body weight for your oxygen consumption or carbon dioxide or energy expenditure. Just use the raw values of mils per hour. Okay, so what is a normal mouse metabolic rate? This was one of the first questions that we were hoping to ask about 5 years ago, and we couldn't really find this because so many people do their experiments in different ways. So June Corrigan in the lab, who's a research assistant, compiled data from the International Mouse Phenotyping Consortium. And this is data from 3,000 mice. And what you see is body mass is one of the biggest factors affecting energy expenditure. So lighter animals, less massive animals, and more massive animals, the larger animals tend to have a larger energy expenditure. Sex here does not appear to play a significant role. In fact, these slopes are not statistically significant, implying that even though there are assumptions that, well, why don't female mice gain as much weight? Is it because they have a higher metabolic rate, or are they more variable? None of that really came out in this analysis. It really just seems like their metabolic rate is smaller than the male mice because their mass is smaller. Another very important factor is temperature. The temperature at which mice are housed affects their metabolic rate very strongly. So these mice are kept in the cold with an ambient temperature of between 18 and 22, and their metabolic rate is much higher than those kept in the warm or sort of at a room temperature of 22 to 26. We quantified that and found that lean mass is the single greatest contributor to energy expenditure. Ambient temperature second. Where they were housed, the institution, is a third. Sex is a small factor, fat mass, and the season. So the statistical approaches for experiments of obesity and thermogenesis, as I mentioned, ANCOVA is extremely useful here, where you can take the mass as the covariate and plot it as this regression plot. There are plenty of data like the respiratory exchange ratio or locomotor activity where mass isn't a factor, and you can simply do a simple ANOVA analysis. That's easy. But in our studies of thermogenesis, we found more and more experiments where these slopes were not parallel. The ANCOVA has as a strict requirement that these two slopes between the groups must be parallel or not significantly not parallel, to put it in statistics speak. So if you have intersecting slopes, you have what's called an interaction effect. And if you imagine this group has more and more brown fat mass and a higher metabolic activity with each gram of fat mass, then it's going to depend on the weight of the animals as to whether or not your effect is significant. The ANCOVA simply cannot handle interaction effects, and so we do something called a generalized linear model. The investigators using CalR, they don't have to know about this, they don't have to care about this. CalR will take the data in and automatically determine whether or not an interaction effect exists and whether we can run ANCOVA or a GLM and just go on with your day and get your results without any user input at all. So when I started running the metabolic core, and even before that, when I was a postdoctoral fellow, to run these analyses, you would get these data files, which would be a file from each mouse, each 10 minutes for each of these variables, carbon dioxide, oxygen, RAR, and you would get spreadsheets that go down thousands of rows and dozens of columns. And you'd have to put that all together into some data frame and then go through and find out where you open the cages and where the data is invalid. And sometimes you have to rescue mice. And this was a data handling nightmare. So it also was in no way transparent or reproducible. I couldn't go back to you 2 months later and say which data I took out when I opened the cage. So this was really not an ideal solution. Together with David Cohen at the Brigham and Women's Hospital, now at Cornell, and Louise Lantier at Vanderbilt, we designed and wrote CalR, which allows you to very simply, and I'll show you an example today, take the mice, the indirect calorimetry data, get these raw data files, read in these raw data files without doing anything to them. You can visualize your data, plot them very quickly, get your analysis by ANOVA, ANCOVA, or GLM, and then get these plots and have your answer. So what really used to take us about 8 hours can now be done in less than 10 minutes pretty reproducibly. So let me show you some of the facts about CalR that go into making it. So in this experiment, we have 2 groups of mice, and we run them first at thermoneutrality at 30 degrees, and then we turn down the temperature, and we're running them at 4 degrees. So in a formal statistical setting, you would take all of this into one statistical model. You would take the mice at 30 degrees in the 2 groups and the mice at 4 degrees of the 2 groups, and you'd calculate whether these 2 conditions at 30 and at 4 are significantly different. So we're not doing that. So you can do that, and surprise, mice at 30 degrees and mice at 4 degrees are significantly different. But that's not what CalR does. CalR will say, this is one experiment, and we're going to analyze whether you have a significant group effect here. And CalR will treat this as a separate independent experiment. And it's done for simplicity because there are too many possible combinations of experiments that you can run for us to have templates to be able to do everything. You can talk to a statistician, and if you want to run to see whether or not mice at 30 degrees and mice at 4 degrees are different, by all means, do that. CalR simply doesn't handle that kind of a problem. So we have a bunch of templates for most of the common experiments. So that is to say, a lot of the experiments that we run commonly in our core at Harvard and our colleagues and our collaborators run, those are the experiments that we can analyze. It doesn't mean that if you've done an experiment that CalR doesn't run, that your experiment's invalid. It also doesn't mean that you can't find another way to analyze it. It's just that CalR is a fast way to do very common experiments, simple experiments, and that's what we have. So the most common one, the one that people use, I'd say 80% of the time on CalR, is if you just have two groups and you want to see, are they significantly different? That's simple. Sometimes people are doing acute responses. You treat an animal with something, or you treat with a intervention, and you want to see over a short period of time, are these statistically different? And so you can get significant calculations at each of the points for a period under 12 hours where you are calculating the appropriate test, ANCOVA or ANOVA, at each of the points for under 12 hours. You can get a 3-group ordered test. So here you have wild type, heterozygote, and knockout mice, and it's a different statistical framework because they are part of this continuum. You can get a 3-group non-ordered analysis. So this is wild type and 2 different knockouts, where each of the groups is really independent from each of the other groups, and 4 non-ordered groups similarly. So you get not only the ANCOVA or the statistical test, but you also get post hoc tests to determine which groups are significant from which other groups. So here's what CalR looks like, and I'll be the first one to admit it is not the most beautiful program in the world. It is meant to be functional, it's meant to be useful, but I'm not an expert programmer, and so I apologize if it's not as pretty as it could be. The other thing that I should point out on the screen is that this is the error that people see most commonly, disconnected from the server. If CalR can't read your file, if it was exported in a way different from the way that CalR was expecting it, the only error that CalR throws back is disconnected from the server. It doesn't mean your internet connection's down. That's the only error message that CalR gives out right now. So what it means is go back and re-export your file. We can read in files from the 3 major manufacturers, from Columbus Instruments, from TSC, and from Sable Systems, and just go back to the instructions and do it that way. Sable Systems and TSC have been very good with their customer support. If you're having difficulty getting your file in there, please contact them. They have very good customer support for their users. So the other thing that you should notice is that you can specify the diets that your animals are using, and it specifies the energetic content. And so this is important for calculating energy in and energy out. For the graphical user interface, we'll also go through this in just a minute. This is the ability to select the appropriate time that you want to analyze. You can select the appropriate variable. You can plot by group or individual. And you can just very quickly read in your data and see, what did my experiment look like? How did it go by group? A lot of the software, we have Acclams, and it's not really easy to know how your experiment actually looked while it's happening or immediately afterwards. So this is very helpful. Louise Lantier helped us a lot with many of the features that went into this. And the last part of the CalR visualization is this analysis table. So it's broken up into 2 parts. The top part is for mass-dependent variables: oxygen consumption, carbon dioxide, energy expenditure, and food intake. So it's plotted as mils per hour, whether you have a significant mass effect and whether you have a significant group effect. So the mass effect, as we discussed, is really whether larger animals have a significantly different rate than the smaller animals. And the group effect is whether there's a significant difference between the groups that you're analyzing. And in here, there's no mass effect for these mass independent things. respiratory exchange ratio has no mass, and therefore you just get the group effect down here. So this is what the mass effect looks like if you look at 3,000 animals. This is a highly significant mass effect where the larger animals have a greater energy expenditure than the smaller animals. And it tells you the relative quality of the experiment. Over here, a group effect is an example of how these mice kept at colder temperature are significantly different from the mice kept at a warmer temperature here. And so that's what people are generally interested in. Are my groups different from one another? CalR has a couple other features that are pretty useful. One is the ability to exclude animals that had problems during the experiment. So this is a real experiment. This is not made up. We ran this experiment, and we took a bunch of mice and turned the temperature down to 4 degrees. I think this is a total of 24 mice. And we saw the averages. So these were the wild types, those were the knockouts, and the hets looked extremely noisy. And using CalR, you can go from a group visualization to an individual visualization. And you can see this one animal, its oxygen consumption dropped precipitously, and then we took it out of the cage so that it didn't die. So through this entire period while the experiment's running, this cage is measuring 0 values for an empty cage. So this here is plotted zeros along with all the other values. And statistically, there's no reason to do that. So if the mouse isn't in the cage and isn't part of the experiment, we can exclude that mouse from the analysis and then proceed on with our analysis. And that's what you can do here in CalR. Furthermore, these calorimetry experiments are very noisy. If you have to open the cage to adjust an animal, to refeed the food, to change the water, a lot of those things can very dramatically affect the results. We see this in RAR more than anything else. Usually it's between the value of 0.7 and 1.0. Opening the cage can give you a value of 3.0 easily. So what this remove outliers feature, it's turned off by default, but you can turn it on. It measures for each group in each photo period whether a particular data point is greater than or less than 3 standard deviations from the group during that time. And if so, and you've turned on outliers, you can remove that one point. So that's from a group, that's from this individual, and then this is after you've removed it. There's also a feature where you can see all of the data that's been excluded and save that as a file, and you can manually curate that to make sure that it was appropriate. So before we go into the examples, just a summary of where the CalR project is now. CalR has been going for a couple years now. Its current statistics say that we've run more than 12,000 sessions. So that's every time someone opens up CalR, and this is only counting sessions that are longer than, I think, 10 seconds or something. Over 4,000 users, most of them in the United States, but over 33 countries. We predefined the statistical approach and cutoffs. So when people say that they use CalR, and hopefully more and more people will keep using it, everyone's using the same, albeit arbitrary, set of standards, the same p-values and the same tests. Nobody has to decide which place to put the star over in their plots anymore because we're all now doing it very similarly. There's a level of reproducibility here because we can export these files, these CalR files, and share them with one another. So after I've run my analysis, I can instantly give it to you, and you can recreate exactly what I've done immediately. You can clean the data sets with this remove outliers function, which saves a lot of time. You can promote collaboration by sharing the data with the shared file formats. Also, the entire website is anonymous. When you open a browser window and you start using it, as soon as you close that browser window, all the information is gone. Everything is kept in RAM, and then when you close the window, it's wiped. People have asked me numerous times, are we saving their data? Are we collecting their data? And I told them, not only are we not collecting the data, but how could I use their data even if I wanted it? You upload 2 groups and you say group A and group B, and what am I supposed to do with that data? It's completely anonymous, and the only information that's collected is the IP address, where the experiment was done, and the duration of the experiment. So CalR is a free open source web tool where investigators can upload their raw data, perform quality control, perform the appropriate multiple regression statistical analysis, and generate publication quality graphs and tables. And you can see the rate at which CalR has been used continues to go up. Okay, so here's an example. This was out of a paper that just was published this week in eLife from my group and that of Owen McGinnis and the Mouse Metabolic Phenotyping Centers. This is an example of a group of mice from the Jackson labs that was placed on either the high-fat diet, 60% high-fat diet, or the research diet's control diet that's supposed to match that. And you can see after about 12 weeks on the diet, the difference between the low-fat diet mice and the high-fat diet mice is the high-fat have gained about an extra 10 grams of body fat. Not very much of a difference in lean mass, and their total mass is increased. So these mice placed on a high-fat diet, they got fat. Why did they get fat? What does indirect calorimetry tell us about that? So you can see their energy expenditure of these high-fat diet mice is higher during the light period and during the dark period. But just like the example that I showed earlier on, this is due to their increased body mass rather than a difference in energy expenditure per se. Their energy intake, which is calculated from the energetic content of the food times the mass of food, looks like it's ever so slightly greater in the high-fat diet mice. Their cumulative energy intake pulls away over time. Not a big effect, but it is there. High-fat diet mice obviously have a lower RER than mice eating a high-carbohydrate diet. And energy balance, so this is energy intake minus energy expenditure, doesn't really look that dramatically different, but ever so slightly positive more positive for the high-fat diet group, with lower locomotor activity for the high-fat diet mice. When you do the ANCOVA and regression plots, you get this sort of complicated looking plot. So if you take low-fat diet mice and you plot them here, this is the regression line, and then you try to extrapolate that, what would be the energy expenditure for a larger mass animal? It would be up here. But these high-fat diet mice seem to have a lower than expected energy expenditure. And part of that is because this extra body mass is not homogeneously lean mass and fat mass like these low-fat diet mice. This is really just added fat mass, which is not very metabolically active, which is why you see this reduced slope of the energy expenditure. Energy intake is slightly more straightforward. They are eating a little bit more, but it's not significant, just like energy expenditure. And in energy balance, the high-fat diet mice are in fact ever so slightly higher, but this is not a significant effect either. These data are noisy. And this brings me to another thing that's been discussed time and time again. It was in the CHOP paper and more recently in another Nature Methods paper, which is the number of mice needed to detect significant differences for energy balance experiments. Assuming that you had 1 kilocalorie per day difference between your group, which is a pretty big difference between your group, and you had a relatively small standard deviation of 1 kcal per day, you would still need more than 16 mice in order to detect that level of significant difference. In this experiment, we're actually seeing a smaller difference between groups, and we actually expect that based on the difference we see in that experiment, we would need probably 400 mice to get a significant difference with that. So wild-type mice on a high-fat diet gain more weight due to gain more weight than the low-fat mice, and they gain an extra 10 grams of fat mass. So they are clearly gaining weight. However, indirect calorimetry cannot detect these significant differences through the measurements that we're making right now, whether it's because the mice are independently too noisy, the machine is too noisy, or simply we just need more mice. It's also possible that we're not accounting for the differences in energy absorption. Fecal calorimetry was not done on these mice, but I think that's unlikely. So it's also possible that the classic P of 0.05 is too conservative for these studies of energy balance. These mice are clearly gaining more weight, yet our indirect calorimetry says none of these effects are significant. So that leaves us with quite a conundrum. All right, so in our example 2, we're going to go online and we're going to use CalR and we're going to see how it actually works. Something you should know about how CalR works is it starts off with this cone where you load in your raw calorimetry data which you can put into a CalR data file, the original unaltered raw data from the experiment. And then you also make a session file, which includes the body mass and composition, the groups, the group names, the group color assignments, the body weights, body compositions. You can specify the photo periods in your animal room. What diet are you using? What time range are you going to analyze? Are you going to exclude the analyzer, the outliers, and are any subjects being excluded? Once you load all of that data and metadata in, then you can analyze and get out the regression plots, the statistical analysis, and the body weight plots. The first time you run this takes 10 minutes to get all of that data in there. However, for subsequent runs, you have your CalR raw data file, and you now have the CalR session file. And the session file contains all of this information, and so you can immediately reproduce your earlier session and keep exploring your data. And this is one of the things that makes CalR really useful, because it's not like the first time you look at your data, you instantly know what it means. We come back to these experiments time and time again as we see more and more things as our hypotheses develop. So let's go over to CalR, and I'll share my screen. Okay. Okay, so we are going to load in this data file. So this is going to be the CalR data file. It's not that big, 1.5 megs. And a session, which we've already made to have all of that data in there. So this is going to pre-populate the light and dark photo periods. It has the weight data, weight and body composition data in it already. It's specifying that both groups are using the same standard lab diet, and we can call these groups anything we want. We can call it blue and yellow. And we've assigned the mice to the group, and you just do that by moving them back and forth. You highlight the animal you want, and you can move it over, or you can move it back. And then you go to plots, generate plot, and you can start seeing immediately what your groups look like. We can change these colors. If you know the hex code by heart, that's great. Otherwise, you can type in those colors, and anytime you make a change, you can generate the plot like that. That's kind of terrible. Let's call that orange instead. So you can see that I've selected only from this time to that time. You might be wondering why. In this particular experiment, half the mice were put in a day early, and those mice were also taken out a day early. So when you look at the whole experiment, you can see those are empty cages through one part, and those are also largely empty cages through one part. So we really don't want to analyze empty cages. So we will just analyze the part that we are interested in when everyone's in the cages. We're looking at oxygen consumption, but you can also see energy expenditure, carbon dioxide, energy balance, the respiratory ratio, food intake, and all the things you get with indirect calorimetry. Let's look at energy expenditure. So it looks very similar to oxygen consumption. You can plot this by the individual animal. When you see large error bars, which we don't have here, you can sometimes identify cages where the lid isn't sealed properly, or there are other experimental problems. None of that's present here. So from here, we can go to the weight plot, and you can see the weights of the two groups in this experiment. You can see the fat mass is similar, the lean mass is similar, and the total mass is also similar. So that's a great setup for a good experiment. If you go over to the regression plots, this takes a moment sometimes. Hold on, weight plots, time plots. Of course, during the live demonstration is when things go wrong. There we go. So you can select the covariate. This would be total mass, lean mass, or fat mass, the time of day, either the light or dark photo periods. And these will calculate on the fly. And it will tell you that you have a significant mass effect, which is not surprising since you have that positive correlation between all mice from low to high body weight, and then the group effect that you have two significantly different groups. These have a significantly higher oxygen consumption, even taking into consideration the body mass. One other neat feature, you can do a three-dimensional plot. This is something that is not always the most useful, but is kind of cool to look at. So you can plot fat mass and lean mass on the two X and Y axes, and then oxygen consumption or any other variable on the Z axis. So that's just kind of a cool thing and helps you visualize your data very quickly. But really the most important is the analysis. So instead of having to look through each of your variables independently, you can get an analysis table that's plotted. So you can see oxygen consumption. While there's a significant mass effect, we're not that interested in the mass effect. It's really oxygen, carbon dioxide, energy expenditure, and energy balance are all significantly different for the 24-hour period, for the light, and for the dark period. These mass independent features, the RER, locomotor activity, ambulatory activity, these are all significant at all of these time points. So these are two very different groups of animals for whatever reason. And so let's go back for a moment. Let's go to Zoom, and we'll go back to our slideshow. Okay, so we now know that it's hard to get statistical significance with normal levels of differences that will give us obesity in mice. So there's a question of, if you have all of these different groups of mice, all these different knockouts, and some of them are going to have a higher energy expenditure, some will be lower, and it's hard to say if using a ANCOVA p-value of less than 0.05 isn't going to be that meaningful because we have biological differences that are not statistically significant, how else can we start to think about this data? So here's just one way that we tried to visualize this, of looking at differences in energy expenditure for some common physiological differences. So if you take young mice, 15-week-old, and you compare them, what are the effects of aging on energy expenditure? So this is a 1-year-old mouse, has a very small difference in its energy expenditure, and then a 2-year-old mouse is almost exactly the same. So aging, at least under the steady state conditions, isn't affecting energy expenditure very dramatically. Exercise, here non-exercise mice, these are given voluntary running wheels. In 1 week, you get a 3 standard deviation effect on energy expenditure, which goes down by 2 or 3 weeks. So this is a transient effect. Mice are really good at thermogenesis, and brown fat is a very powerful activator of metabolism in mice. And if you take mice at thermoneutrality and inject them with a beta-adrenergic agonist, CL, you can see a huge effect, more than 0.2 kcals per hour. This is similar to the effects that you see if you take a mouse at room temperature and you either decrease the temperature to increase the metabolic rate, or you increase the temperature to lower the metabolic rate. So we can start to put these things in context, and we can struggle a little bit with the statistical significance. We further went on to do an analysis of more than 2,600 knockout strains. I apologize, my neighbor is outside cutting his yard. We did analysis of more than 2,600 knockout strains for the International Knockout Mouse Phenotype Project. And what we can see is most of the mice do not have a very different energy expenditure between the knockout and their appropriate wild type groups, but some fraction of them do have 1 standard deviation less or more, 2 standard deviations less or more, or 3 standard deviations less or more. And you can kind of see that in this plot, where some of them, despite the mass, still have very different changes in their energy expenditure. So our future plans are to develop a repository for indirect calorimetry, which we're calling creatively the Cal repository. We'd love for people to be able to take their data that they run in CalR and be able to put it as part of this database. In the database, they could then add to the knowledge with all these other 2,600 or thousands and thousands of other strains, and you could see what the relative effect size of your knockout or your intervention was compared to all the other ones that have been published about mouse indirect calorimetry. So to summarize, the best practices for analysis of obesity and energy balance, to show regression plots to see mass dependence. So this still is not that common. We've been doing it. Some other people have been doing it. You can get a post hoc test to show what's significant or not, but this is far more instructive than the bar plots. The analysis of ANCOVA or GLM is much better than the t-test that you'll see for the bar, and you really do have to take mass into consideration for these mass-dependent factors. If at all possible, analyze groups before body weight or body composition has diverged, and use body composition data to increase precision if possible. Never ever plot per body weight or, God forbid, would be one of these exponents when you're comparing the same strain with just differences in body composition. This is really, it's wrong. Try to share your data in a repository or as supplemental data. And CalR is a solution to the problem that fits many, but not all complex scenarios. Just because we don't do, CalR can't help your problem doesn't mean that yours isn't valid. We just haven't gotten to it yet. And I'd like to acknowledge Amir Mina, who spearheaded CalR. He was a research assistant in the lab. He's now a MD PhD student. June Corrigan, also a research assistant in the lab, who's done our large-scale analysis. Deepti Ramachandran, Yuchin also helped quite a bit. Katie got this started. Essential collaborators, David Cohen at Cornell, who had the first idea to start this. Louise has been a tremendous help in validating and experimenting with this model. Owen McGinnis for developing the MMPC reproducibility data set, which we based a lot of our analysis on. And we've had help from the manufacturers of the indirect calorimetry systems to help us be compatible with the systems and have as few problems as possible. We've received support from the NIDDK, from the MMPC, the micromouse projects, and for an indirect calorimeter, as well as from the Harvard Digestive Disease Centers. And I hope you guys found that helpful, and I'm happy to take any questions. Thank you.
So Alex, there are some questions coming from chat. Let me give you the question. So first one is from Heidi. Heidi said, I've tried using CalR previously, but wasn't successful in uploading the data file in CalR. Does it have to be in a specific format when extracting the data from the system?
Yes, that's a great question. It has to be in a very exact data format. So there are instructions on the CalR website under help. So each of the three different systems works very differently: the Sable system, the TSC system, or the Columbus instrument system. So they all have different requirements, and you have to follow different steps to do it. But we have very few problems with the Columbus instruments system. We have fewer problems with the Sable system, and we have considerably more problems with the TSC system. They are working on an upgrade of their software, their PhenoMaster software, and they're expecting to have a lot of that solved very soon. Reach out to them, the manufacturer, and if that doesn't work, then email us. But it does have to be a very specific file format.
Okay, thank you. So the second question is, can we see the exact format of the example data files?
Yes, I can show you those now, but so for our CalR publication and also on our website, we have these data available to download. So the supplemental material from our 2018 cell metabolism paper, Mina et al., contains all of the data files, including the example that I used. That is already there. You can download those data files. And then the paper that we're publishing this week in eLife has more CalR data files, including that one of the low-fat versus high-fat diet mice that don't reach statistical significance. Those CalR files are there as well. So there's a lot out there that you can get started with on your CalR journey.
Okay. The third question is coming from Jocelyn. Can we export the code used to generate plots?
Yes, so the code used to generate plots is part of CalR, so it's not particularly modular. So if what you mean is, can you export the data? Yes, there is a feature, and let me go back to sharing my screen on CalR, and I can show you exactly where that is, because that's a great question. So if you go to your time plots over here, so all the data that you've selected based on whatever these hours are and the groups that you specified, you can download data averaged by hour, and then you get the data, all the data grouped by blue or yellow or whatever your groups are per hour, and then you can plot that however you like with your own system, GraphPad, or if you prefer Python to R, however you like to plot things, you can get the data out from CalR and then you do your own analysis that way. If you're interested in getting the daily averages, so under the average plot, there's also this button right here, download data average by day, or the overall averages, which are further down here. You can take the data averaged overall and plug that into your favorite ANCOVA program, because that really has already done the hard work of all the data curation.
Okay, so the next question, can you combine runs? For example, if you repeat the experiment?
Yes, there is a tool on CalR up at the top for combining runs. If you go here, and then combine runs is just down there. So we can do that as well.
Okay, next question coming from Adil. Thanks for the useful overview of the program. I have 3 questions. So first one, how long of an acclimatization period would allow the mice to have? The second one is there a way to combine data from separate runs? I think it has been answered. The third one is there a way to pull numerical data from the problem that was used to generate graphs so that we can make our own graphs for publication?
All right, so I think I've answered 2 and 3. So the first question is, how long an acclimation period would we allow the mice to have? So we typically do less than 24 hours, but usually more than 6 hours. So the best answer, I mean, there's no correct answer. What we typically do is we want, if they were started in the middle of a photo period, we'll give them the rest of that light or dark period to finish their acclimation. And then when the lights shift, they typically start their normal pattern over again. They're finished exploring the cage and they're ready to get down to living in the cage. That's not always hard and fast and true. Older mice sometimes take longer to adjust. High-fat diet mice take longer to adjust, but generally less than 24 hours, I think, is adequate acclimation.
Okay. So next question coming from Mafada from Stable System. One of the most common questions we get from customer is about the analysis of food liquid choice experiment. Is this planned to be handled by CalR?
No, we haven't planned on doing food choice or liquid choice experiments. That's just not something that we're set up to do right now. But Stable Systems could always develop that kind of software. They're welcome to do it. So most of the calorimeters don't have that kind of hardware system in it. And what we're looking to do is making something that everyone with any calorimeter can use and be useful most broadly. But that sounds a little bit too specialized for what we're doing right now.
Thank you. So next question from Elizabeth. I'm using Stable System and Stable System's newest software. The mouse labels are 1_1 or 1_2_1_3. How do I change this?
You know, I don't know about Stable Systems' newest software. I think we have to talk to Stable Systems about that particular feature. In fact, the previous questioner could probably answer your question. So maybe you could meet up and talk about that. That sounds like an issue with the Stable software.
So one question from Daniel, is CalR an R package?
So CalR is an R package, but it's not publicly available yet. We've debated this over and over again, do we make CalR open source? Do we allow people to run their own versions of CalR, make their own additions to CalR, and potentially alter what it does? And for the moment, having it as this web repository where if you want to run CalR and have CalR analysis, you've run it through this web server that everyone else is running at the same time. So we've held off on making it open source, but that is something that we've been debating back and forth the pros and cons. If you can talk me into the merits of releasing it into the wild and then having to support that as well, maybe you can, but so far it seems like more of a downside than an upside.
Okay, next question from Jennifer. Where can we find historic data such as from a repository that will show indirect calorimetry data of mice from different genetic backgrounds?
I don't know that repository exists. I'd love to make it, but I don't think that's there. I mean, what we have is a lot of people from all over the world running different experiments on their own systems, but usually in their own formats. And I think if we could get a repository, hopefully sponsored by the NIDDK or NIH or NSF, we could have a repository where we could start to build exactly what you described, Jennifer. And that'd be so useful, where we could say, here's what C57 mice look like, and here's what CD1 mice look like on these diets with these temperatures. And we could start to actually understand the normal variation in mouse metabolism. But that doesn't exist yet, but that's a great, great point.
So next question from Heidi. At the Jackson Laboratory, we are running extremely large studies with multiple groups and longitudinal time points. Is there a plan to expand the capacity of CalR to compare more than 3 groups and calculate batch effect? How many runs can be combined for analysis now?
So we've never had an upper limit of mice. I think we've done 64 mice just because that's all that we had. We are working with a group at the NIH that's doing a very large longitudinal study. And the solution that we came up with is, rather than trying to plot everything, so you can read the data into CalR, but ultimately you do that single day, the total overall analysis for energy expenditure, for oxygen consumption, for all the metabolic variables. And then you put that into a giant table. And then you're going to run the ANCOVA. So it's going to be at each time point, you'll compare all the animals and all the metabolic variables at that time point using the ANCOVA table. So CalR, in that sense, isn't really going to fit what you need to do for plotting, but it could help you to read in all that data, get the answer that you want, and then help to build another table that you could then plot in R or some other tool. And I'd be happy to talk to you about what we did with the NIH for that. It was a little bit labor intensive, but it solved the problem that they had. But that's a really great system. And I'd love to see your data if you're willing to share.
Okay, I have a last question from chat. What is the interaction effect statistics mean exactly?
Right, interaction effects. So it's an interaction between the mass effect and the group effect. So the mass effect, as I described, is smaller mice will have a lower metabolic rate, or whatever the variable you're measuring, let's say it's oxygen consumption, than the larger mice. Okay, that's the mass effect. The group effect is that the two groups are significantly different. And then the interaction means that those two factors are interacting so that under certain masses, the effect will be significant. And then under certain other masses, it wouldn't be significant. So it's hard to have a biological interpretation. What's the physiological meaning of an interaction effect? My answer is that it's going to be complicated, and it's going to depend on the mass of your mice as to whether or not it's significant. But those are the statistics. And so the thermogenesis studies are really complicated because of that reason.
Okay, 2 more questions.
Okay.
Okay, so one from Ivana. If you have an experiment where you change the temperature during the course of the experiment, for example, cold room temperature, thermal, do you break this into 3 different data files before uploading for analysis?
Oh, that's a great question, Ivana. So we have the CalR data file, which will be the entire run, which will have the 3 different temperatures. And then we'll have 3 different session files, which will just highlight those temperatures at which you're analyzing at the cold, at the room temperature, or the thermoneutrality. So we'll have 3 3 separate session files, which you can then go back, just load your data file, the session file for the thermoneutrality, and then run your analysis whenever you want. So that's the easy way of doing it, and that's very fast. We do that all the time, and I think that's a really good solution.
One question from Jennifer. Have you thought about the pitfalls and advantages of combining the DSI telemetry with indirect calorimetry?
Yeah, so I don't know if she knows or not, but we just received a grant from the NIH 2 weeks ago in order to combine the DSI telemetry to measure glucose with our indirect calorimeter. And because we have this CalR infrastructure already present, we thought we could just overlay the DSI glucose telemetry. We can also do body temperature telemetry at the same time. With our existing system, we've been able to combine body temperature, which has been very useful and very fruitful. But the glucose is going to be very different. And I think being able to say glucose, what is the glucose level combined with the RAR level and the food intake all at the same time is going to be really helpful. I don't exactly know how well it's going to work. I don't know what the pitfalls are going to be, but within the next 6 months, we should have the answer to that. If you have any experience, I'd love to hear if you've already tried something and you know something and I don't, but we're just going to dive in with both feet.
Okay, one question from Caroline. Thank you, Alex, for informatics. Regarding the statistics we provided, does the group effect mean that the groups are different, even accounting for mass?
Yes, that's exactly what it means. You've got it, Carolyn. So the group effect means that your groups are different, even when you account for the masses of the group. So if your group effect is statistically significant, then you can report in your publication that these groups are significantly different.
Okay, 14th question, can I get the presentation slide? We will share the slide and the recording video through dkNET channel, and I will send a link to everybody. Yeah, and there's another question, maybe it's a comment from Jocelyn. For open source, the same defaults can be specified in an R package that are specified in the GUI. Making it open source will also allow users to expand applications, add new experiments more rapidly to meet users' need. Good luck, this is awesome. And from next question, and if you don't have body composition, can you have confidence in your data?
So if your groups are very similar in body weight, in overall body weight, absolutely, you can have confidence in your data. Even if your groups are a little bit divergent, it's probably okay. It's when you have very big differences in body weight, you kind of need to know where those differences are. Do you have a more muscular, less fat, larger mouse, or is it all adipose tissue? There are ways of doing it where you would dissect out the mice after sacrifice and try to calculate back their percent body fat. It's not great. In the old days, before people had these MRIs, we used to saponify mice to determine their total body fat content. I do not recommend that, but it can be done even without technology. So keep your mice similar in weights, and then you don't necessarily need body composition data.
Okay, I don't see any more questions in the chat.
Great. Well, thank you everyone for your participation. I'm glad we had so many people here today.
Yeah.
And feel free to reach out and email me if you have more questions.
Yeah, so if you have more questions, you can email Alex directly, or you can contact us and I will forward it to Alex. And thanks for Alex for the presentation, and thanks for everyone joining today's webinar. Like I mentioned before, a recorded version of this webinar will be available via dkNET YouTube channel. We like to encourage you to share today's webinar with your colleagues and students. Our next webinar will be on May 15. It's next Friday. And we will do a live demo on dkNET Hypothesis Center Signaling Pathway project. Dr. Neil McKimmon will show how you can generate hypotheses using consensus transcriptional regulatory signatures, and will give you a live demo on how to identify genes and signaling pathways in cellular response to coronavirus infection. This tool is designed for bench scientists. No programming skills are required. To receive upcoming webinar information, please subscribe to our newsletter, follow us on Twitter, Facebook, or check announcement at dkNET.org. Thank you again and have a great weekend.
Thank you very much, Kowei.
Thank you.
Bye-bye.
Bye.